Hello, dear students. Today we are going to discuss the gene regulation, taking into consideration the lac operon. Okay. Before we discuss the lac operon, let's know what is the operon. Operon means a group of structural and regulatory genes working together. Okay. Jacob and Monod in 1961 studied this lac operon on Escherichia coli. Escherichia coli is a bacterium found in the colon of the human beings and other mammals. It uses usually the glucose, okay, for energy production, for survival. When there is no glucose, it shifts its off operon into on condition. Okay. Let me tell you people what is regulation. You have the fan and you have a regulator which regulates its power supply. Okay. When you need the large amount of air, then you regulate the regulator. You switch the regulator so that a large amount of electricity or the power is provided to the fan and it runs faster. And when you want the slow fan, you switch the regulator and the fan would be slow. It would work slowly. And when you don't need the air, what would you do? You would switch it off. Okay, so you regulate it. So regulation means either production of something. If you are taking into consideration certain gene, it means you want to produce something, or you want less production, or you don't need the production of that very protein. Okay, here is what I have divided the environments into two conditions. One condition is when glucose is available. When glucose is available, what happens? The bacterium uses glucose for the production of energy, and no other source is required. Okay, right. So what happens? Glucose is used. It is broken down. It is catabolized, and ATP are produced, and its respiration takes place, and bacterium lives. Okay. On the other hand, this is that condition. Okay, and lac operon is switched off. It is no more needed. Okay. As far as the structure of lac operon is concerned, as I have mentioned earlier, that the operon is a group of genes which consist of structural as well as regulatory genes working together. So here is that very gene, which we are a group of genes which we are going to discuss today. Here is the regulatory gene. This is promoter gene, P for promoter. Here is operator gene. And these three are the S1, S2, and S3. These are the structural genes. Okay, they work together. But they would work. This is lac operon. This is lac operon. Okay, a group of genes working together in which the promoter gene and operator and structural genes, they work together. Here is a regulatory gene. It also regulates them. Okay. Now suppose that there is uh, an ample amount of glucose present in the environment of bacterium. Here is what glucose which is present. Okay, it needs no more other source of energy, so it would utilize glucose. So if it is using glucose, why it should operate certain other source of energy? That would be futile. So the regulatory gene switches it off. How? It has actually two parts. Its regulatory gene has two parts. One is promoter part, and the other is inhibitory part. The inhibitory part produces messenger RNA. The messenger RNA expresses itself. This is what messenger RNA. Messenger RNA. It produces, or it expresses itself in the form of repressor protein. Repressor protein does its function in such a way that it would bind here at the operator site. Okay. When it is bound here, there would be no more space for the RNA polymerase. And if RNA polymerase cannot form messenger RNA, then the messenger RNA won't be able. If there is no more messenger RNA, then there would be no more production of the proteins. There would be no more production of enzymes, and those enzymes are not being produced. Why? Because nature has devised that the cell may not exhaust itself for the futile action, right? So gene S1 and S2 and S3 won't express themselves. Clear? Now, here is an environment. This is an environment. This environment is there when the glucose is available and the bacterium is living very happily using glucose for the production of energy, and no other source is required. Okay. There is another condition when glucose is no more present, but lactose is present. Just suppose that glucose is not present in your intestine, and you're taking what milk. You know what is the milk protein? Milk sugar, that is lactose. And lactose, consists of two monosaccharides. Those are what? Galactose and glucose. Let me write the structure of what. Rather, I have written here. You see, this is the structure of galactose, and this is glucose. They have joined together to form what? Lactose. It is the milk sugar. The milk sugar, as it has a glucose molecule, it would be furnished if here it is cleaved, it is broken down, it is hydrolyzed, and that hydrolysis can occur only when some enzyme is produced by the genes of this bacterium. So what happens in this condition? Here is what? The operator site has been suppressed. The inhibition has occurred. Here the inhibitor protein is being produced. The inhibitor gene, this is O, okay, and it produces messenger RNA. Messenger RNA expresses itself in the form of a suppressor protein, okay, suppressor, right. Now, if lactose is present, suppose this is the lactose molecule, okay, if lactose is present, lactose would join with this suppressor protein. Suppressor protein suppresses operator gene, okay. Now, if it is present, it would rather suppress the suppressor itself, okay. How? Here is this phenomenon taking place. Now, this suppressor protein is no more able to suppress the operator gene. When it is free, what happens? Here would be the joining of what? This is, suppose, RNA polymerase. The RNA polymerase would be happy now. Okay, it is happy now. Okay, it is happy. It would act. It would express itself. What is produced? The structural gene first are Z gene. It is some authors they say that it is Z gene, or you can dub it you can name it as S1. Sorry, it produces beta galactosidase. Beta galactosidase. What is its function? Its function is to, suppose this is what this is glucose and this is galactose. It would cleave this molecule, and glucose would be furnished, and that glucose would be utilized for the production of energy for the respiratory process by this bacterium. If lactose is needed, say this is lactose, suppose, okay, this molecule is lactose, this has been utilized. Now, new lactose should be taken up by the bacterium, okay. If here are present the lactose molecules, no more glucose molecules, these should be taken in. This is, say for example, bacterium, and this is bacterium. Suppose this is bacterium, E. coli. Here are present the lactose molecules. These molecules should be taken in. The plasma membrane should be made permeable. So permease enzyme is required for taking up the lactose molecules. So here the S2 gene produces what? Permease. What? Permease. And the third one is a gene which produces what? Trans-acetylase. Trans-acetylase. Trans-acetylase has been hypothesized to help these two to facilitate particularly the permease enzyme, okay. Now, what is the stimulus? What is the inducer of this on system, are making this gene system or the Operon system functional? What is the inducer? The inducer is what? Lactose. So lactose is here an inducer substance for what? Lacoperon. Taken? Now, if there is less amount of glucose, say here there is large amount of glucose, okay, the bacterium would happily live. As I have mentioned earlier, it will keep on functioning. It would enjoy glucose, okay, it will produce energy and live. But if there is lesser amount of glucose, then the bacterium would feel glucose hungry, and it would be now knowing that there is something wrong. So it would produce a substance which is known as cyclic AMP. What? Cyclic AMP. So here is adenine, okay, this is the ribose sugar, and this is what? Phosphate, okay. 1 2 3 4 5, okay. Phosphate is attached here with what? This ribose sugar. And this cyclic AMP, along with another protein named as catabolite activator protein, or CAP protein. What? CAP protein. They both together activate what? The promoter site of what? The gene which acts to utilize glucose from what? The Lacoperon. So this is the inducer, and this inducer in turn is activated by what? Catabolite activator protein, and it is activated by cyclic AMP. So this is how the beautiful system of molecules has been organized by nature in this very small organism. Okay, I think you should have understood well the Lacoperon here through this discussion. If you have understood well, you must share this video to your friends. Thank you very much.